I will talk today on network models in biology and in a few applications to molecular machinery, and then predicting missense variance and chromosomal dynamics. So here is the plan. As you can see, we have 3 sections. Elastic network models, I will just give some basics. That part will be very short, assuming that many of you may be already familiar, or if you are interested, you can learn easily. And then I'm going to talk about 2 important applications. One is how we can understand whether a missense variant or a mutation in a protein is harmful, is pathogenic or not, using information from elastic network models, actually, in addition to sequence and structural data. And then I'm going to talk about chromosomal dynamics. Actually, you know, we used to think of the chromosome as a sequence of 4 letters. This changed completely in recent advances in technology, characterizing the structure of the chromosomes. And now we are able to use our models to understand. I think there is somebody who is not on mute. Okay, so I start with elastic network models. So we have, as you can see on the left, this is just a very pretty image showing how nodes and springs would be connecting the structural units in a network model. So in our model, we have the most commonly used would be the nodes representing the alpha carbons. And here you can see how using the elastic network models, we are able to visualize the types of equilibrium motions that take place, specifically at some interfaces where the interactions are critical. Now, here's just a brief summary. Suppose you have a given structure of protein. Here is HIV reverse transcriptase. What we do is we map it to an elastic network representation. And a close-up view is just a system of beads and springs. A very talented student of mine created this movie, which I can show briefly. So you have a given structure of a protein here at late kinase. We focus on the alpha carbons for each representing each amino acid shown in spheres, and we connect those within a certain distance, usually 7 to 10 angstrom. We got our network model. And then we use classical theories like normal mode analysis to understand what are the equilibrium motions of this protein. Why is it important? Because we think protein motions define function. So here again, for that HIV reverse transcriptase, now we have a spectrum of motions, actually, we call modes. This is mode 1, mode 2, 3, 4, etc. And the mode number increases with the frequency of the motion and decreases with the size, if you wish. The mode 1 is the most cooperative. As you go to higher modes, they are stiffer and they have smaller amplitudes. They are more localized. So elastic network models are very useful because they take account, rigorous account of contact topology. So the contact between residues that form a protein. So in this issue of Science, the whole issue was dedicated to understanding how form, the overall topology, the shape, dictates the underlying forces and under equilibrium conditions, which in turn define the equilibrium motions. So we are able to dissect the mode spectra and understand cooperative events that the structure uniquely stabilizes. I'm going to present today the application to those 2 applications, the chromosomal dynamics and the pathogenicity of single amino acid variants, subs. So I would like just to show the type of results we get usually from elastic network model analysis. For example, here, the chaperonin, GROYEL, some of you may be familiar with. This is the side view, this is the top view of the molecular machine. And indeed, this chaperonin helps fold unfolded or misfolded proteins in its central cavity. And the way it operates is like a washing machine. You have two counter rotations of two cylindrical entities, and in the cavity in between, you have the proteins are being processed to fold. Or you have a membrane protein here, AMPA receptor composed of 4 monomers. And this is a schematic representation of the membrane, which even the membrane fluctuations, undulations are coupled to the motions of the protein. And again, we believe if we know the structure and we would like to understand the function, we need to predict the intrinsic dynamics, and elastic network models are very useful. And they are applicable not only to crystal structures or NMR structures that are deposited in the protein data bank, but now you may have heard about CryoEM Revolution these days. So we can use electron density maps and then construct those types of structures, again, network models, to understand how the big machines, like here, the mammalian chaperonin, cooperatively move to achieve their function. So for those of you who are interested, there is an interface ProD which you can use. Usually you can enter a favorite protein sequence, for example, and it's going to give you all the proteins in the data bank that are homologous or to this protein, and you can define the sequence identity threshold. And then you can do an ensemble analysis of existing data, or you can do an A&M analysis of a representative member and compare the results, the mode shapes, or you can compare with much more expansive MD simulations. Essentially, that is an interface that allows to turn those static images of proteins into animations, showing what are the most accessible motions. So that interface has been used by more than 150,000 unique users and more than 2 million downloads. I'm really surprised to see how many people are interested in protein dynamics. As a parenthesis, the first student here, the student Ivet Bahar here, was a graduate from Koç University. And I had the pleasure of mentoring him as a PhD student of mine. So he really made a big difference by creating that interface. He's now working as a senior researcher at Google in San Francisco. Now, this is another interface for biologists or those of you who are not skilled computationally. And everything has been designed here to facilitate, the usage, to give you a pleasant experience. Again, you can enter your favorite protein. That has the advantage of also constructing the environment, for example, the membrane for membrane proteins. So that is another interface I would suggest you to use. So that's all about the elastic network models. I'm now moving to more recent applications. So the first one I would like to emphasize is how we can predict the functional impact of missense variance using elastic network models. A paper we published a couple of years ago, and last year we also implemented the interface. So this is actually a combination of physical science and engineering and machine learning methods. So again, we're focusing on single amino acid variants. So we have variants all the time. Some of them are hereditary, others are acquired due to aging, cancer, etc. in our DNA. The mutation takes place in the DNA. Now, what happens is that in the DNA, some of the nucleotide substitutions, they are which are called missense variants, result in a change in amino acid type. So that is an example here. A T replaced by an A gives rise to a protein amino acid change from aspartate to glutamate. So now we would like to understand whether this mutation is neutral or deleterious. As a matter of fact, actually, most of the mutations are inconsequential. Most of them are neutral. And but then if when there are such variants, we really want to know whether a disease in a patient is caused by a mutation that's not neutral. So what we do is we calculate a pathogenicity score. And in principle, you would like that pathogenicity score to be such that it would provide a clear discrimination between neutral and deleterious substitutions. So if all the neutral substitutions give you this histogram, the probability distributional scores, and all the deleterious give such a, so except for a region, narrow region, you can really very clearly discriminate them, correct? But that's not the case. That is what the real situation is here. Look at those two histograms. It is really, at first, quite discouraging to see how those two distributions overlap. And now it gets really hard to start to discriminate whether a given mutation is really deleterious or neutral over quite a broad range of pathogenicity scores. So what we wanted to increase, the amount of information for deriving this type of scores. And what we did is the following. Most of the approaches in the literature use sequence information. Polyphantry is one of them. And a little bit more advanced approaches started to use structural information. Polyphantry, again, is one of the most broadly used tools. So you start with a non-synonymous single nucleotide polymorphism in the DNA, and you would like to see in the sequence, protein sequence, what's happening. So they would be looking at sequence alignments among homologs to see whether this is a position that is conserved, in which case you don't want any mutation there. You assume that it's deleterious. But then we also look at surface structure properties. For example, if the mutation is on the surface of the protein, it's going to tolerate substitutions because of the spatial constraints. So using this type of learning from existing data, everything being fed to a probabilistic classifier, we are able to make predictions. And we know the confidence levels as well, predictions about the whether a mutation is going to be deleterious or neutral. This is a binary outcome, very simple. And we can visualize also in 3D. Now we said, okay, this is the state of the art. This was the state of the art in 2018. Now we're going to add a new feature, dynamical features. So based on the elastic network model, fluctuations, perturbation response scanning analysis that Atılgan and Atılgan started at Sabancı, and mechanical bridging, etc. A bunch of properties. And now we are going to train a random forest classifier using 20,000 human variants that have been annotated. Annotated means we know whether they are neutral or deleterious. And we would like to see whether adding this type of features, dynamical features, and even more structural features helps us improve our predictions. Now, the first thing in such analyses, of course, is to have a data set. And we, in the literature, when we look at the data set, you can see here how, there are many, many data sets on human mutations, variants, and not all of them are distinctive. There are overlaps, etc. So we took all of them and generated an integrated data set of 20,000 unique single amino acid variants, which have known structure, because we know we need the structural data for the wild type or a homolog, not necessarily the mutant, in order to make some inferences. And then when we did that, and then we checked how we predict in this type of tenfold cross-validation, you hide 90% of the data, you try to predict the remaining 10%, you keep shuffling, etc. So when we did that, we could see that you may be familiar with this type of RAC plots for false positives versus true positives. If everything is random, if our machine learning algorithm is not predicting anything, so depending on, of course, the distribution of neutral and deleterious, if they are 50-50, we are going to obtain this line normally. But now if we are predicting something, just using sequence, this is the curve you obtain, and just using structure and dynamics, this is the other curve. I'm sorry, structure and dynamics is the other curve. But if you use all three properties, you can see already how much better we do compared to random predictions. So that was very encouraging. And then this pair of histograms here, based on the sequence, you can see that they already improve if we take structure and dynamics into consideration. But if we take all of them, all three properties, now we are in a much better situation. So at this level, for example, these predicted scores can be mapped to probabilities. Here you can say that if the score is 0.1, it's 100% neutral. If the score is 0.8, it is there is that much probability of being neutral or deleterious, etc. So we got this type of histograms, which we used for making predictions. And then we added also more recently a new feature, sequence coevolution. So coevolution, because we were inspired by this work of Chris Sanders and Deborah Marks at Harvard, where they show that really mutational effects that are predicted from sequence covariation are very powerful in discriminating what is good, what is bad. So we added that feature, and then we showed that our prediction accuracy is better than existing tools. So the paper has lots of details. For those of you who are not familiar with what I'm talking about, sequence evolution or coevolution, this is the concept in a nutshell. You just take the sequences, you align them. Here is your what's called a multiple sequence alignment. What are those sequences? These are the sequences corresponding to your protein of interest and its orthologs and paralogs. So across different species or family members in the same species. And you would like to see at every single position, suppose at this position, what you see is it is all variant. We have 20 different types of amino acids here. They are all conserved. And you can calculate using information entropy, Shannon entropy, what is the entropy at this position. Higher entropy means it is not conserved. Lower entropy means it is highly conserved. Or you can start also look at pairs, as you can see here. So the position I and I plus 5, for example, each time the arginine is replaced by an glutamic acid, you have also a glutamic acid being replaced by a lysine. Remember, these R is positively charged as well as K. These are negatively charged. So there are what we call compensating mutations. So if the protein family is able to undergo this type of compensating mutations or amino acids that co-evolve with others, coevolution, then we can also have a metric, mutual information, that will tell us the probability that such a compensating change will occur and the information content, which we could use in our interface that we developed. So we have included all those properties to generate this new interface, Rhapsody. Rhapsody, the name is Rapid High Accuracy Prediction of Single Amino Acid Variance Outcome based on dynamics. It took us a long time to find this acronym. Now, what does Rhapsody do? Suppose you have a mutant, you have a protein in which you have an amino acid substitution. Now you can just enter the query, and then it's going to give you an output about, what is the probability that this mutation is going to be impacting negatively or positively the function. And we also generate this type of saturation mutagenesis in silico heat maps. So what you have here is the residue index for a given protein. And the y-axis is the 20 types of amino acids, one of which is the wild type amino acid colored white. And all others are possible mutations, okay? Now, this diagram tells you at every single position what is going to be the consequence of replacing the wild type amino acid by others, 19 types. And the color code will tell you the probability of being deleterious or neutral. If it is blue, it is going to be neutral. In the case of red entries, you have deleterious. So this type of color-coded map can guide you as to which sites are really very hard to mutate, those red regions. Whatever you do here, you replace the wild type, it's going to be bad. It's going to negatively impact the biological function. Or here, this is quite tolerant, as you can see, to mutations. And then also we have more specific data on specific substitutions. So by the way, when we did that, we found out that in the literature, there was about 5,000 variants, which were not included in the previous step for us, when we were training our machine learning classifier. And then we tested again our algorithm against these. And again, the accuracy level is for sequence and dynamics and structure, all of them is 0.82. So 82% of time we can predict accurately. If we were using sequence only, it's 74%, dynamics only is 67%. And that is also a comparison with several other methods in the literature. Some of them are biased because of the input data set. And we can see that our classifier has been outperforming them. Okay. Oh, here is an application. Now that we have this tool, we've been applying to many, many different proteins. One of them is ROMK, which is involved in kidney functions, potassium processing. And our collaborator, Jeff, he asked about a certain, 15 variants that has been observed in patients, and whether this could be related to Bartter syndrome observed in those. These are Bartter syndrome patients. And then we were able to correctly classify 13 out of these 15, 13 as either neutral or deleterious, but we made correct predictions. And then he experimentally, after our predictions, he experimentally tested their behavior in yeast grow assays. And indeed, that's how we concluded that, okay, the predictions were accurate. So that saves a lot of time. Now you can do a first screening, and if you would like to learn more, you can do experimental tests, of course, but that is a very useful tool for personalized medicine in particular. Okay, so now in summary, by including dynamics, in addition to sequence and structure, we can make a better assessment of the impact of mutations. Elastic network, we could use the elastic network models for that because they are very efficient. We can serially apply to thousands of mutants. And what is beautiful is that it's not a black box. We know exactly why a certain mutation is predicted to be deleterious, because now we have many properties as a function of residue number. And now we know that, for example, this position may not be deleterious because it has a very high solvent accessible surface area. It's very exposed. So we can rationalize the observations based on the features that we have been predicting and including in our classifier. So these are, for example, whether the residue serves as an effector or a sensor of signals allosteric signals, etc. So there are many features. And future steps, we haven't done actually, we haven't explicitly incorporated the change in the neighborhood of an amino acid when it undergoes those conformational changes. And some changes in neighborhood could be really serious. It could give rise to, disruption of function. And for example, in this case, it locks the protein in an inactive form because it finds a new partner that the protein likes a lot, and it's not going to go back. It's not going to do its back and forth motion, which is necessary for function. Or here we can apply to complexes, multimers, assemblies. That's also still missing. So we would like to include in our algorithm information on the protein-protein interactions. So something may be surface exposed, which is good, but if that surface exposed residue is involved in an interaction with another protein, you don't want to mess with it. And then finally, everything here was just based on uniform parameters of elastic network models, but we could include more specific parameters for interactions and further we can sharpen the results. Now I'm moving to the next item, chromosomal dynamics. So we will have the questions at the very end. Chromatin plasticity, this is a very new concept actually. Again, as I mentioned, we used to think of the chromatin or the 23 chromosomes that form the chromatin as a, information factory with purely defined by the sequence of nucleotides. That's not true. Now we've learned that the structure matters, and the structure, not only the double helical DNA, but the histones, nucleosomes that form, or the way everything is being wrapped up in the nucleus. And they call that chromatin plasticity, which defines the cell fate and identity. We don't like the word plasticity because a plastic material will undergo a change that would be irreversible. It's going to flow. We like better elasticity. Otherwise, our DNA cannot function. It needs to have elasticity. And at some point with aging, indeed, it becomes more plastic. Okay, so here's another view. This is a great review by Tom Mistelli at NIH. Again, showing it is more than a sequence now. It is a cellular entity, and that show, there are some regions. You can define many structural elements at different scales. So gradually you have more and more nucleotides forming chromosome domains, compartments, and everything is squeezed now in the nucleus. And interesting, they call that chromosome territory. All the 23 chromosomes that we see here are, they're not intertwined. They have their own territories in the three-dimensional space. So the inter-chromosomal contacts are usually very few, only the surface, but most of them are intra-chromosomal. So this, we wanted, again, we say that the structure is always related to function. Now we have information on structure. How do we have information? This is high-C data. I'm going to get back to that. But we have information on gene-gene contacts. That's what we have. And then knowing this type of contact topology between different genes, the protein encoding regions and even non-encoding regions of the DNA. Now it is possible to examine their dynamics, to our view, to gain insights into the basis of function, transcription, etc. So we are going to do that. So we've started to work on this topic with Carl Kingsford at Carnegie Mellon, who is a computational genomics person. That was not my area, but I brought the methodology. And the first paper we published in 2017. So it's very new, but the field is very new as well. The experimental data are just in the last 5-6 years, perhaps, or at most a decade. Now we have chromosomal dynamics that we predicted by an elastic network model in one paper. And more recently, we've been further pushing the idea to understand how cells are distinguished in terms of their chromosomal dynamics. And here are two very talented students from my lab who have contributed to those studies. So we use the Gaussian network model, the simplest elastic network model. So what do we do? The data from experiments is called high C data. Originally it was 3C, 3C for chromatin conformation capture. Then now they changed to high C, it is easier. And what those data, they give us information, these contact maps between gene loci. So this is the entire, for example, chromosome, one of the chromosomes, or it could be the chromatin. And the diagonal elements will be the nearest contacts, right? And the off-diagonal elements would be those separated sequentially. So we have this type of contact map between gene loci. What is a gene loci? It is a segment of the DNA that usually contains 3-4 genes. So we have information on protein coding and not coding regions. It is interesting because, you know, as you look at the three-dimensional structure of the DNA, if you enlarge one part of it, you know, the way it is structured is reminiscent of the organization at a higher scale. So there is some kind of scaling, which some of you may be familiar with the fractal geometry of nature by Benoit Mandelbrot, who wrote the first book on that, actually. So for example, if you look at fern, this plant, and you can see that these sections, leaves themselves, repeat the entire plant. And you could even focus on one smaller portion here, and it's going to repeat the entire thing. So you have the same pattern that repeats itself at different scales. So that is what we can do also with network models. We can construct them at multiple scales. So we can think of a network node representing an atom, a residue, a rigid block, an entire domain, or even a gene locus now, in the case of application to chromosome. We're talking, a gene locus is about 5,000 kilobase. Think about it, 5,000 nucleotides. This is huge. Now we are condensing everything there into a single node, but that's good enough for observing many, many features, as you're gonna see. Now, the idea is we take the contact map, we turn it into what is known our Kirchhoff or connectivity matrix. Essentially, this is identical more or less, but we have some normalization to clean the noise. So a few technical stuff. And then you do an eigenvalue decomposition. You obtain eigenvectors for those of you who are familiar. But the bottom line is that you extract the dominant features, cross correlations between the gene loci or their fluctuations in space. So for each gene loci, now we have a certain mobility. We call that mobility profile. How do they move in three-dimensional space, right? So that we have compared this with experimental data, two types of experiments, ATAC and DNase, SEC, where we have the locus here, each point representing 5,000 kilobase pairs, and the predicted mobilities by mean square fluctuations of those loci by the GNM in blue, and the other two are the experimental data. And you can see that even though the methodology is very simple, we already have good agreement with very expensive, very time-consuming experiments. And then this was for one of the chromosomes, and we could do this type of analysis for all of them. And you can see that the comparison with ATAC and DNase-seq, two experimental sets versus theory, and the comparison between two sets of experiments, they are comparable. But actually, the comparison with DNase, this red one, is the best. It even agrees better than the two sets of experiments in between. So we have, and then we can also, probably I will not go into details, but we can also look at cross correlations, which were measured by ChIA PET experiments. In this case, they call them long range, but they are separated by 100 kilobase pair. So it is not the diagonal is autocorrelation, but very close to the diagonal, those red points, which they have identified to be cross-correlated. Then we said, okay, we're going to take another, control set, to pairs of gene loci separated by the same distance, like the background, and see whether those pairs identified by ChIA PET to strongly correlate are really more strongly correlating than the background. And that's what we observe here. You can see that those experimentally identified to be covarying in terms of their gene expression patterns. High C data were also found experimentally. Were also found in our calculations to be covarying in space. That is very meaningful. Now we are relating gene expression patterns to 3D features, and the backgrounds pairs were not correlated. So now there is also this concept I kept showing, this multi-scale representation from, it's multiple scales from nucleosomes all the way to the chromosome territories, you remember. And there are TADs, topologically associated domains. So we would like to see whether this type of organization is universal, the same in all cells, and what is different. We know that cells, in all cells we have the same DNA, but not all proteins are expressed in all cells. It depends on the specific tissue functionality of the cell. So then we would like to trace this type of different functions of the cells to their chromosomal dynamics. So we analyzed 16 different types of cells, as you can see here, and then plotted here their fluctuation spectrum for different types of cells. And you can see there are differences when we look at cross correlations in this heat map between different cells. Well, there are, some of them are similar, others are not. And here, if you take every single column and take the average, so how much, for example, that particular cell HSPC correlates with all others, so that is going to give you this type of violent plots. So you have, on the average, the level of correlation is around 0.63 between different cells. And for example, those skin cells or hair, they are very different because they are very stiff, and you can see they distinguish themselves. So this difference, 0.63, so the cells are not identical. Okay, their dynamics are different. The different dynamics may originate from two sources. Either they have access to different types of modes of motions, or they have the same repertoire of motions, but their frequencies are different. Okay, so when we did this type of analysis, indeed we found that you see how now the curves, they all became almost identical here. The only thing we did was to eliminate the differences due to frequencies. So the same, again, ensemble repertoire of motions are available to all cells, but some of them are more probable than others, depending on the cell type. So this reminds us of the fact that all cells have the same DNA, but not all cells express the same proteins. So then again, we said we may relate this type of changes in mobilities or frequencies to the expression, the synthesis of proteins. Now, again, this is summarizing what I just said. The cells, they enjoy a bunch of motions here. They jump, they run, if you like. But not all modes are deployed in the same way. Some of the cells, they like to jump, others, they like to dance. And those gene mobilities is what defines the cell identity. and cell identity is relevant to its function. So we said that we're going to look at the, in a given cell type, for example, what are the most highly mobile genes, HMGs, we call them. And now we can, it is better to subtract from the mean to, magnify the signals. So we have this type of curve, and we know where individual genes are located. So we know that this gene is highly mobile compared to all others, or let's say this gene is very rigid. And then we said, does high mobility of the genes correlate with its upregulation, with its high expression level? So if it is highly mobile, does it mean that this cell is going to be expressing that highly mobile gene? Okay, so we have made this type of comparison. We know which ones are highly expressed in different types of cells. So we have cell type 1, cell type 2, 3. In this database, for each chromosome, we know the protein gene expression patterns, the transcriptomics, if you wish, RNA-seq data, for those of you who are familiar. Now, we're going to just go through this filtering procedure where we have, for example, the mobility profile for one of the genes, one of the cells, HMG. And we would like to see whether this particular filter is gonna discriminate among those unknown cells, which one is indeed IMR90. This is so this high mobility profile curve here for IMR90, are we gonna recognize that? And then we find that indeed one of the cells shows a very high correlation. And then when we look at the identity of the cells, we indeed see that this is IMR90 fibroblast actually. And if we rank order them, these cells, unknown cells, based on their similarity of their gene expression profiles to the gene mobility profiles, we can see that this rank ordered listing will accurately capture the cell that we used as our query. So overall, we can see that we have now been moving forward with understanding 4D genome. 4D means the structure and its dynamics. So these are your 23 chromosomes in, well, it's from a mouse embryonic stem cell. And this is the way for a given structural model, the way all these chromosomes move concertedly. So we've seen good agreement with many different types of data. And it was very interesting to find out that some modes are silent depending on the cell type, and highly mobile genes are highly expressed. So the dynamics, in a sense, defines the cell identity. Just a discussion, the analysis, everything refers in the AIChE data that we use. Everything refers to a population of cells. These are average behaviors that we have seen. And now there are also more recent experiments with single cells, and data from single cells may provide even more accurate information, not only between different cell types, but heterogeneities among cells belonging to the same type. So that is, and then we can, use all these methods now to also better understand non-coding regions that regulate transcriptional activities. So with that, I would like to thank you for listening. And I was very lucky. I have been very lucky to have excellent collaborators and lab members. So this photo is a little bit, this one is a little bit old. That one is more recent. So this is the way we meet these days. And here, the chromosomal studies were mainly performed by John. Daniel was involved in Rhapsody development, and all other members are, contributing to our ongoing activities. Thank you very much. So I'm going to stop sharing. Okay.
Thanks so much for your presentation. We can take your questions if you have any, or you can just raise your hand.
I hope it was not too you know, different for your background. I came from your background, but I became a computational biologist.
May I ask a question?
Sure. Yasemin.
I don't know, just my name, Yasemin Yaşaroğlu.
Nice to meet you.
Nice to meet you too, Hocam. Thank you for, how can I say, coming. You mentioned the interpretability of the deleterious genes, and we know that if they are deleterious and something like that. And I want to know, do you have problems with machine learning models, the interpretability problems?
We do. We have lots of problems, actually. And I didn't have time to discuss, but it's good that you raised the question. So what happens is that, you know, in those databases, they give you the binary information. They say that it's, you know, it's not gonna impact the function, they say, it's neutral. And then they say it's or deleterious. Now, deleterious is a very, very broad word, in the sense that it is actually the real, you know, specification is that it does affect function. And when it does affect function, there may be in some cases a gain of function or a loss of function. For example, the protein may be doing more what it's supposed to do, and that is considered to be deleterious. But in some cases, actually, you would like the protein, in some diseases, you would like the protein to do more. So the information in those databases from which we learn is only binary. So they say it's good or bad. But what they say bad is not necessarily bad always. So we really need shades or more specificity on the types of diagnostic they write there. So it is, for example, what we would like to do is to move to a, instead of a binary classification, to a three-way, to say, okay, it is deleterious, but this deleterious behavior originates from a gain of function or loss of function. We want to discriminate that. So we are very limited by the information that is available in those databases. And we believe that with growing data, our classifiers are going to do better. So it can only get better. That's a good thing. We keep learning from accumulating data. But right now we are limited. And even now, you know, we have 82% accuracy for this binary classification, which is not bad, but we would like to be predicting more.
Thank you.
Good question. Good question.
Can I ask something, Bahar Hocam?
Sure, sure.
Yeah, I'm Berkay. So I was wondering, how do you define the mobility of these chromosomes from the high-C data? So in principle, we are cross-linking the DNA, and then we are making it the rigid, like a rigid structure. So we are observing the last conformation that this chromosome has. So how can we define the mobility from this data?
Yeah. So, you know, the data, you are right. There is a cross-linking procedure, and I had a slide on that, but let me explain. So in the DNA, you are cross-linking two strands that are close in space, right? But then you are chopping the two ends and then ligating those two short segments, right? So you had these two things making contacts, cross-link them. You chopped both of them and you ligated one of them. And now you have a small stretch composed of two segments that were spatially neighboring each other. Okay. Now, the information, the data they give us is, they tell us, okay, that particular segment, and that they can identify what these segments are. They say these two segments now are in the same, small region. And so they were originally making contacts in space in 3 dimensions. So the information from this type of cross-linking and ligation experiments, and this also happens at regions that are usually exposed, as you also mentioned. So the information tells us, they tell us which particular segments, not necessarily connected, maybe distant neighbors, but spatially close, make contacts. So they give us contact topology.
Yeah, so basically we are seeing the contacts between one region of DNA and to the another region to the DNA.
These are called the loci, one locus and another locus. Yes.
Okay, so like can we mention some elastic features, something like NMR versus X-ray, for example? We know that the X-ray is like a crystal structure, but the NMR is in a solvent. So we know that this structure is is not rigid. But when we have these contacts from AIChE data, we have the like X-ray version of DNA. Let's say. So like
we don't have the X-rays.
Yeah. We don't have, but like when we get the structural information from the proteins, we have the some rigid structure from X-ray, and from AIChE also. I mean, I don't know if there is any particular assay to get such structural information, but again, like when we are saying mobility, like it has to, I think, move a bit.
Yes. So the thing is, Berkay, we can't measure what the overall chromatin structure is. And the thing is actually that there is an ensemble of conformations. It's moving a lot, actually. It's fluctuating everything. Even though they are confined to some territories, as I mentioned, there is an ensemble of conformation too. But we have indirect information. We have information on which part of the chromatin is exposed. We have information on which gene loci make contacts. So these are pieces, little pieces, bits and pieces. Now, using this type of constraints, which ones make contacts, it is possible to construct a structural model. That's what NMR scientists do. They have those chemical shifts and NOE factors, etc. And using those data, they build models. But you don't usually build a unique conformation. You have an ensemble of them. And what is happening with the chromatin is, again, you cannot resolve a single structure for many reasons. You cannot crystallize, it's too big, etc. But also, it is not itself uniquely defined. It samples multiple conformations. So but what we can do is that, knowing this ensemble, we know in this ensemble which part really move a lot. So we are, in a sense, skipping the 3-dimensional description, and we are gaining information on the fluctuations in this 3-dimensional structure. I've shown a movie, for one chromatin from the mouse genome. However, that is based on one model, and there are multiple models, structural models. I'm not sure if I answered your question. The beautiful thing is that contact topology allows us to calculate the fluctuations, and that is the fluctuations, the mobilities, they define also the function.
Yes, thank you, Hocam.
Thank you. Okay.
Question, yeah.
Yeah. So I want to ask a question about Rhapsody. So are there any patterns in mutations that Rhapsody cannot predict correctly? Like, are there any class of mutations that Rhapsody is not very good at catching, or are they distributed randomly?
So, you know, that is a good question too. So the methods that we are using, you know, when they are only based on sequence, they are limited. You know, they are not sufficient usually. And if suppose you don't have any structure, we can't know anything about the structural effect of the mutation. So we don't know its effect on the dynamics. So we will rely fully on sequence alignments. But then the bottleneck is also to have a sufficiently large number of sequences. So those multiple sequence alignments where you see, you know, I showed how a given position may be conserved or co-evolving. If this alignment doesn't contain, let's say, at least hundreds, but often we want thousands of sequences for that protein, then we cannot also make accurate predictions. So it all depends, again, on the available data, sequence data, structural data.
Okay.
And there, you know, some proteins are disordered, you know. The disordered ones, they don't have structure, so we cannot apply those methods. And these are really hard to predict. They are already exposed and disordered, and we cannot discriminate easily different sites. Yeah.
Yeah, we have one question in the chat. I will be reading it for you.
Dennis?
Okay. Yeah, okay. Arda is asking how far away are we from genetic engineering for the entire human genome or DNA?
I'm sorry, I couldn't hear. Could you repeat?
How far away are we from genetic engineering for the entire human genome or DNA?
Oh, that's you know, people are making progress a lot in genetic engineering. Those CRISPR experiments, last year, the Nobel Prize was received by a scientist doing that. So it is, there is a lot of progress, but you know, you don't want to engineer the entire genome either, right? But if there is a region that is responsible for a disease, there you would like to make a modification. And there is a lot of progress. Again, as I mentioned, even last year's Nobel Prize by Jennifer Doudna, I think she's at UCSF or Berkeley, I don't remember. But that was all about gene editing. So you can edit the genes, you can make changes. So there is a lot of progress. And now, you know, these type of methods that we're developing are also useful for letting experimentalists know which particular substitutions are going to be okay or which ones you don't want to do, etc. So that is important. It's a pleasure to see Özlem.
Yes, my apologies. I just couldn't make it.
You made it. You're here.
Yes. I invited everybody, and then.
You know, Özlem is my daughter. Did you know that? Yeah, she is my academic daughter.
I'm very happy. I'm very proud of you.
Well, I'm very scared. You can ask a very difficult question. You're not asking anything.
And my daughter is here as well, Nurcan.
Hi, Nurcan too. You know, Nurcan, we would like to expand Rhapsody to take account of protein-protein interactions. And this interface prism that you developed, I think it's very powerful. Do you agree?
To whom was that question?
I'm asking to Nurcan and Özlem.
Yes, I think, yeah, we discussed about it, I guess, through WhatsApp, right? But it's a very doable thing, and we should do that.
We should do that. You know, it is really important to be combining
multiple disciplines, multiple tools. This is the only way we can tackle those complex problems. And I think you've done a fantastic job by designing, developing the PRISM software.
Yeah, I mean, now it's been up for almost 15 years.
Yeah.
Oh.
So it is time to further use it in personalized medicine now in our applications. That would be great.
Yeah, we would love to.
Okay.
We discussed it with Attila as well. So hopefully
we should do that.
Yeah.
We should definitely do that.
Currently we used your ProD.
ProD, yeah, everybody is using ProD and also making changes. So I really, what Ahmet did is phenomenal. And we just published the third version in bioinformatics. It appeared. The problem is they accepted it within a week or so without change. I wanted to make some changes, you know, to improve the paper. They didn't allow me to touch it. It was theirs. So they have, so that also shows how much ProD is really well received in the broader community. We are very happy. And I would strongly recommend you chemical engineers and statisticians and biologists to try to see if you could use it. It is very useful.
Yes.
It has many features also.
Many features, and it's modular. So
exactly, we keep adding new features for pharmacology, drug discovery, or allostery, etc. Yes.
We have just sent the paper for roots issue, the special issue that I'm editing.
Okay. I should, right? Why don't you send me an email?
Okay. we used ProD for our paper.
Great. So any questions other than scientific from the students? Any career questions? You're welcome to ask. Otherwise, I'm gonna leave. Just if you have any questions, I'll be happy to answer.
You can ask your question.
So I have to say, I don't know if you've seen in the news, last year I was very happy to be elected to the National Academy of Science, but this year there is another Turkish lady. Did you know that?
No.
She has been elected yesterday or the day before. We voted actually, and I'm so happy. Her name is Serap Aksoy. She's at Yale. We have the same age. She's also from Istanbul, and she is a microbiologist actually, focusing on rare diseases. Serap Aksoy from Yale. So now this is the second Turkish. Yeah, I'm so glad. I look forward to meet with her, but we are all at home for the time. Yeah, that happened 2 days ago. So you may, take a look at her work, but this is really a great accomplishment of hers.
And yours as well.
Well, mine is not, now this is Serap. This is her work.
Very, very proud for us.
Yeah.
We're very proud of you. So Stan is a question, I guess?
Yes. First of all, thank you for your presentation. I will ask, what do you expect from students who are just starting to work with you? I mean, for example, I come to your lab, say, Hocam, I want to work with you. What would you expect, or what should I do to prep myself? Because even though I understood the basics of your presentation, I am completely lost on the specifics. I don't know if it is natural or like I'm.
It is very, very normal, okay. So don't feel disappointed. It's extremely normal. But if you have followed some part of it, that is good. I'm not sure which level you are talking. Are you a PhD student? Are you interested in joining as a PhD student or after postdoctoral fellow?
No, I'm an undergrad.
You're an undergrad, and you are probably then talking about PhD studies, right?
Master's or PhD, I guess.
Yeah, actually.
I don't know. If you want to do research, usually researchers, they do their PhD studies, doctoral studies. And then, so to do that, you need to apply to our program, PhD program. We have a joint program with Carnegie Mellon. You may know that Carnegie Mellon University is one of the best in computer science. We are one of the top 5 institutes in as the School of Medicine in the US. And then we have this joint program that benefits, mutually both institutions. So computational biology PhD program that I founded in 2005. So this is a great program. Unfortunately, it is getting a little bit too competitive to get admitted. If you are accepted to the program, then you can choose whomever you would like to work with. You can work with me, you can work with anyone you like. For postdoctoral studies, it is easier. If you have a PhD already and if you have the right background, you just write to me. There is no admissions committee. I just evaluate and get back to you. But in principle, when there is an applicant, let me tell you what is my most important criterion. I believe anybody can do research. You don't have to have extraordinary skills, talents. But what you need to have is a strong desire to do that. It's all about your level of enthusiasm. You have, then you become more energetic, you get more interested. To me, the most important criterion, which I would like to see in anyone, joining my lab as a researcher, is their strong desire to do so, their level of enthusiasm. Does it make sense? So to me, that is the most important thing. And anybody can do anything if you really want it very much. That's, I believe in that.
I completely agree.
Yeah. So yeah, I've seen many people who are very intelligent, but not so interested, who don't perform as well, as good as well as others, who may be less talented, but really very much into it. So it is a matter of being genuinely interested and strongly wanting to do that.
And you shouldn't give up, right?
No, yeah,
keep on
persistence.
Yes, persistence is also.
Persistence. And even more than persistence, I started to use the word patience also. You need to be very patient in the very beginning, because this area is not so. In the very beginning, you may submit a paper, you may get rejected, or you can fail, your program doesn't work, your experiments fail, etc. Many things happen. But it is, but if you are patient enough to do one more time, you will succeed eventually. There is no other way you will succeed. So that is all. That's.
Thank you.
Thank you. This is, many of us learned this the hard way. Nobody told me that the most important thing is to really like what you're doing and want to do what you're doing. But I've seen this, among students, researchers, those who really are very excited, enthusiastic, who have a passion, to do that. Okay.
I was lucky to see you as the role model.
Oh, thank you, Özlem. But I think now this is the new generation. And I also believe in all of you. You have, since you are already at this university, you have achieved something.
Yes,
but that's not the beginning. That's, then you have already the potential to move forward. One last thing I want to say, again, if you want to do research, most of our graduates right now, 2/3 of our graduates, they're not in academia. They go to industry, to pharma, to biotech. Ahmed is, for example, working for Google, as I mentioned, and I have 3 other students working for Google. So there are lots of opportunities in this field, and they are not necessarily academic. So that is a very rapidly evolving field. And those of you who are interested, if you have any questions, feel free to contact me, okay?
I think it's also the same here.
Is it?
At least half of our graduates are now in the industry.
That's wonderful.
One is in.
We need. Yeah, that's great.
They're working as data
really,
scientists.
we need scientists. We need researchers in industry. That is great. But of course, as an academician, I'm very happy to be an academician. Don't misunderstand. All right. Any other questions, scientific or non-scientific? Thank you very much.
Oh.
It was a pleasure, and I wish all of you good luck.